Now come to the grouping of the viruses on the basis of epidemiological criteria. Separate from the formal universal taxonomic system and the formal and vernacular nomenclature that stem from it, there are other categorization of the virus that are practical and useful. Then these are based on the virus tropism and mode of transmission. Most viruses of the animal are transmitted by inhalation, injection, including via arthropod bite, close contact, including sexual contacts, or congenitally. Enteric viruses are usually acquired by the ingestion, fecal-oral route transmission, and replicate primarily in the intestinal tract. The term is usually restricted to the viruses that remain localized in the intestinal tract rather than causing generalized infection. Enteric viruses are included in the family Picornaviridae, इसमें जीनस एंटरोवायरस है. इसके बाद कैल्सीविरिडी, एस्ट्रोविरिडी, कोरोनाविरिडी, रिट्रोविरिडी में रियोविरिडी में जेनेरा है रोटावायरस एंड रियोवायरस. इसके बाद पार्वोविरिडी एंड एडिनोविरिडी. रिस्पायरेटरी वायरस, अभी एंटरिक वायरस की बात की, अब रिस्पायरेटरी वायरस. रिस्पायरेटरी वायरसेस आर यूजुअली एक्वायर्ड बाय द इनहेलेशन, respiratory transmission or by fomites, जैसे inanimated object carrying virus contagion. Fomites मतलब होता है inanimate objects carrying virus contagion and replicate primarily in the respiratory tract. The term is usually restricted to the virus that remain localized in the respiratory tract rather than causing generalized infection. Respiratory viruses included the family of Picornaviridae, जिसमें आता है rhinovirus genus. इसके बाद कैल्सीविरिडी, कोरोनाविरिडी में जीनस कोरोना वायरस. इसके बाद पैरामिक्सोविरिडी जिसमें जीनस पैरामिक्सोवायरस, रूबेला वायरस एंड न्यूमो वायरस आता है. देन ऑर्थ्रोमिक्सोविरिडी एंड एडिनोविरिडी. आर्बोवायरसेस फ्रॉम आर्थ्रोपोड बोर्न वायरसेस रिप्लिकेट इन देयर हेमेटोफेगस यानी कि ब्लड फीडिंग आर्थ्रोपोड वेक्टर and are then transmitted by the bite to vertebrate host, wherein virus replication process produces viremia of sufficient magnitude to infect other blood-feeding arthropods. Thus, the cycle is perpetuated. Arboviruses are included in the family like Togaviridae, Flaviviridae, Rhabdoviridae, Baniaviridae, Rioviridae. इसमें रियोविरिडी में जेनरा है ऑर्बोवायरस एंड कोल्टीवायरसेस एंड अस्फाविरिडी. Oncogenesis virus are acquired by close contact including sexual contact, injection, fomites and by unknown means. The viruses usually infect only specific cells in the particular target tissue, whether they usually become persistent and may evoke transformation of the host cell, which may in turn progress to malignancy. Viruses that have demonstrated the capacity to be oncogenic in the experimental animal or in the nature are included in the family Retroviridae, Epidemiological, Papoviridae, Adenoviridae, and Herpesviridae. Now next is the taxonomy and the causal relationship between virus and diseases. One of the landmark in the history of infectious diseases was development of Henle-Koch postulates, Henle Koch postulates, which established the evidence required to prove a causal relationship between a particular infectious agent and a particular disease. These simple postulates were originally drawn up for the bacteria but were revised in 1937 by Rivers, Rivers, and again in 1982 by Evans, Evans, in attempts to accommodate the special problem of providing disease causation by viruses. A special problem of proving disease causation by viruses. ये टेबल 2.3 में आपको मिल जाएगा. In many cases, virologists have had to rely on indirect evidence, guilt by association, guilt by association, with association based on epidemiologic data and pattern of serologic positivity in the population. The framework of the virus taxonomy again play a role, especially in the trying to distinguish an etiological rather than a coincidental or opportunistic relationship between a virus and a given disease. For example, early in the investigation of the human acquired immunodeficiency syndrome, yani AIDS, before its etiology was established, many kinds of viruses were being isolated from patients and may indicate etiologic agents were being advanced. Prediction that the etiologic agent would turn out to be a member of the family Retroviridae was based on the year in the veterinary research on animal retroviruses and animal retroviral diseases. The prediction was based on the recognition of common biologic and pathogenic characteristics of AIDS and animal retroviral diseases. This prediction this prediction guided many of early experiments in the find to etiologic agent of AIDS. Later, after human immunodeficiency virus HIV was discovered, its morphological similarities to equine infectious anemia virus, a prototypic member of genus lentiviruses, family retroviruses, was the key. Just a minute, I am taking another page from the book. Again, I am telling about the things that after immunodeficiency virus was discovered, its morphological similarity to equine infectious anemia virus, a prototypic member of the genus lentivirus family Retroviridae, was the key to unbelievingly confusion over the fact that the human virus killed host lymphocyte rather than a transforming them as typical oncogenic retroviruses would do. Ever since, the essential quality of comparative medicine has been guided HIV, AIDS, experimental design in many areas, including drug design, diagnostics, and vaccine development. Thanks for listening. Chapter number 2 from book Murphy Virology.